Hi, my name is Kelsi. I am a first-year PhD student at the School of Medicine, and today I will be talking about my project, which is overcoming estrogen receptor-positive therapeutic resistance. Slide, please. 1 in 8 women will have breast cancer. A majority of breast cancer is the estrogen receptor-positive subtype. To treat this, hormonal therapy is used. However, patients are becoming resistant to the hormonal therapies. As a result, a new drug class is being developed called SERDs, but the way they're being tested preclinically is flawed. To imagine this, I want you to imagine the body as a garden. Weeds are like estrogen receptor-positive breast cancer. The SERDs are like weed killer, and when developing the weed killer in pre-market research, they're only looking at it in terms of the weed, not the entire garden. The same thing is happening in preclinical approaches when testing SERDs. The treatment is isolated to breast cancer cell lines alone, which is not representative of what's happening in the patient's body. A literature review has revealed that there are 13 SERDs in development, but only one that's FDA approved. And this is because the preclinical environment lacks 2 aspects. It is the cancer environment and the resistance seen in the patients. So the environment we consider by a large component of breast tissue being adipose or fat cells. The fat cells can influence how cancer behaves. As such, in my model, we want to develop a co-culture of fat cells with cancer cell lines to better represent the environment that the cancer is growing in. The second aspect to address is the treatment-resistant side. This will be done by introducing the co-culture to increasing levels of the traditional hormonal therapies to establish the resistance. And finally, we want to identify patterns of resistance in these models by single-cell RNA sequencing. This would be like taking the cancerous weeds and the fat flowers and blending them into a smoothie. And our super special blender can taste the smoothie and deconstruct it into different components like roots, stems, leaves. The same can be done with our co-culture models. And from these subgroups of cells, we can identify what exactly is causing the resistance and keep this in mind when developing these new SERDs. So not only can we develop better SERDs preclinically, but also in the patient. Thank you.